So we are outside of McCullough Building, which is one of the main centers for material science in Stanford campus. And we will be meeting with Professor Melosh, who works in material sciences as well as biotechnology and who has really interesting projects going.
Good afternoon, Professor Melosh. Thank you for having us here. You're a professor here in the Department of Material Science at the Stanford University. Could you first briefly explain what material science engineering is?
So material science is really the science of designing materials for particular applications, synthesizing new materials, characterizing those properties, and then relating the structures that you see during synthesis to the actual properties, and really understanding at an atomic level how materials behave and how to improve them.
How do you feel that material science could give a unique solutions for today's and tomorrow's biotechnology problems?
So this is really a unique approach to biomaterials and biology interfaces in general, because it really looks at how you manipulate things, inorganic objects, let's say, at the atomic level, and then seeing how you can integrate that into biology is something that's been tried, I would say, in a broad sense before, where you just modify things kind of uniformly. But now with today's technologies, you can really start to design and engineer structures at the nanometer level. So I can design something in an inorganic material that matches the same size scale as an individual protein. And that will give us kind of control that we haven't had before for interfaces between the two.
Experiments or research in the field of biotechnology. Could you talk about one of those experiments?
Sure. So we have a couple different projects that look at the interface between inorganic materials and living cells. And so some of the things that we're really excited about are trying to, for example, measure what is going on inside a neuron as it fires. And so the really key thing to measure there is the electrical properties, and then particularly the voltage across the cell membrane as it is doing its business and firing action potentials and even collecting subcellular or subthreshold signals from other cells. You want to measure all of these little events and then see how that correlates with the larger scale behavior of the network. So what we do is we actually make electrodes that can stealthily insert themselves into the membrane, and that will give you a channel that goes across the cell membrane and lets you measure the voltage from inside the cell to outside the cell. That's really the critical thing, without completely disrupting the cell membrane. As you can imagine, you can apply voltage and you can basically rupture the membrane, but of course neurons don't like that terribly much, and it disrupts their native behavior. And so what we wanted to do is actually design an inorganic metal electrode to look just like a transmembrane protein. So in a transmembrane protein, they have a hydrophobic surface that actually causes them to insert into the lipid bilayer right there. And so we replicated that on an inorganic material, in this case a metal stack, with just a 5 nanometer high band of hydrophobicity. And that enables that to stealthily insert into the lipid and give us nice electrical connection from inside to outside, but not disrupting the cell membrane. So these cells are healthy for longer, we can stimulate and record from them quite nicely, and then we can monitor their behavior over a long time.
How do you foresee relationship between material science and biotechnology?
I definitely think you're seeing them starting to come much closer together. Traditionally, the materials side has been in the realm of tissue engineering, where you try to mimic a tissue with materials, which is still a thriving and ongoing field. I think one of the hallmarks of the last 10 years you've seen in biological technology, though, is really seeing that mechanics of, say, the substrates start to matter. So if you put different cells on soft substrates, they behave differently than on hard substrates. So cells really do sense their mechanical environment quite a bit. That fact is becoming appreciated more and more, and I think as we see how much the cell's behavior is really regulated by mechanical forces and things that are not just chemically related, you're going to see more materials processing and ideas introduced into the system.
Thank you very much, Professor Melosh.